Today I'm going to talk about Dr. Jewel Plummer Cobb, the woman who breaks filters. Dr. Jewel Plummer Cobb was an African American biologist and cancer researcher. As an African American woman, she faced obstacles during her pursuit of higher education. In one of her famous articles, Filters for Women in Science, Cobb compared the selection process of a scientific career to a filter, the pores of which were smaller for women than for men during her time. Dissatisfied by this inequity, she devoted her entire life to not only avoiding being stopped by the filters herself, but also fighting for changes that break these filters in the scientific world and in higher education. Cobb came out of a somewhat economically privileged family. This allowed her to engage in intellectual exploration during her early life. Her mother, Carabel, was a physical education and dance teacher, and her father, Frank, was the first African American to graduate with an MD from Cornell University, who later became a dermatologist. Cobb joined UMich in 1942. During that time, African American students are forced to live in segregated housing. Unsatisfied with segregation, she transferred to Talladega College in Alabama, a private and historically black liberal arts college. But this also meant that she had to start all over again, since Talladega College did not accept transfer credits. Nevertheless, she still managed to graduate within 3 years with a BA in biology in 1944. During her time there, she was also part of the Alpha Kappa Alpha sorority, which is the first intercollegiated African American sorority. After graduation, she joined NYU, despite being initially rejected due to her race. She was later granted fellowship and eventually obtained her MS degree in 1947 and PhD in cell physiology in 1950. After graduation, she worked at the National Cancer Institute at Harlem Hospital from 1950 to 1952. She serves as the head of the biological laboratory at Sarah Lawrence College between 1960 and 1969. She became a corporation member of the Marine Biological Laboratory in 1972. Now let's talk about Cobb's contribution to biochemistry and cancer research. Her research topics involve studying cancer chemotherapeutic agents both in vitro and in vivo. In particular, some of her works focus on understanding the relationship between in vitro and in vivo responses to chemotherapeutic agents. In addition, she was also an expert at manipulating melanocytes. In one of her work, The Comparative Cytological Effects of Several Alkylating Agents on Human Normal and Neoplastic Cells in Tissue Culture, she uses HeLa cells as models to understand how 5 different alkylating agents cause changes in cancer cell mitosis. Alkylating agents, defined as compounds that contain no less than one alkylating group, are known to cause damage to DNA molecules. In particular, they do so by forming cross-linkages between strands of DNA. In this study, the molecules of interest are mustargin, triethylene melamine, triethylene thiophosphoramide, also known as thiotepa, chlorambucil, and phenylalanine nitrogen mustard. Notice that 3 of these molecules share a common functional group, nitrogen mustard, which is known in the biochemical world for its alkylating properties. Cobb discovered that all of the 5 molecules are capable of stopping mitosis in neoplastic cells by causing damage to chromosomal structures. Later in 1961, she published another paper. In this study, she tested 3 of the molecules that were tested in the previous paper. In addition, she also tested actinomycin D and methotrexate. All of the tested molecules are capable of inhibiting mitosis and eventually causing cytolysis of neoplastic cells. One of her in vivo studies was a case series study where Cobb investigated 5 chemotherapeutic agents for disseminated carcinoma of the breast. This time she tested 5 compounds, 4 of which already appeared in her previous works. The new molecule here is purimycin, which is an aminonucleoside antibiotic. Nucleoside refers to this nucleoside component on top, and amino refers to this amino acid at the bottom that looks similar to a tyrosine. Because of this molecule's resemblance of amino acylated tRNA, it is capable of causing premature termination of translation. However, the, res- the result was unfavorable to some extent, demonstrating that only thiotepa and methotrexate have some effect as a treatment of carcinoma of the breast. Now you might wonder, how do in vivo and in vitro responses of human cancer cells to chemotherapeutic agents relate or differ from each other? To answer this question, Cobb published a paper in the prestigious New England Journal of Medicine in 1957. In this case series study of 37 cases, she observed a somewhat strong correlation between clinical responses and responses of tissue culture. As a conclusion, Cobb concluded that instead of using animal screening as part of the drug discovery process for chemotherapeutic agents, it might be more plausible to use human cancer cells culture for screening, especially given the parallel between clinical and tissue culture responses demonstrated in this work. Lastly, she was also an expert at manipulating melanocytes. After 1960, Cobb spent much of her time studying human melanoma cells. An important contribution Cobb has brought to the realm of melanoma research involves the discovery of treatment with melanocyte-stimulating hormone in combination with cytokeratin B as a way of helping melanoma cells retain their pigments in vitro even after a long periods of cultivation. This technique is important since allowing melanocytes to retain their pigments in vitro for longer periods of time allows a better study of melanoma in vitro. Among the figures shown to the right, figure 1 and 3 show melanocytes before and after treated with melanocyte-stimulating hormone. Figures 2 and 4 further show that adding cytokeratin B enhances the effect of melanocyte-stimulating hormone by allowing the melanocytes to grow even bigger. In addition to her scientific achievements, she also devoted herself to promoting equity and representation for underrepresented groups. One of her most famous contributions is the post-baccalaureate pre-medical programs for minority students. This program was first established by Cobb at Connecticut College and later inspired similar programs at other institutions. It turns out that enrolling minority students in this program ended up facilitating a virtuous cycle. In particular, as more minority students get enrolled in this program, more minority students will get accepted into medical schools. And as the statistics looks better, more minority students are encouraged to consider medical school and healthcare as their future careers. She also devoted her efforts to addressing the issue of the underrepresentation of females in science. During her address at the Women in Engineering Conference in 1991, she described the life of a woman scientist as going through a series of filters where most women scientists face obstacles during their pursuit of science, and only a few of them eventually come out of the filters. The filters she mentioned in her address at the Women in Engineering Conference actually referred back to an earlier article she published back in 1979. She claimed that the minimal representation of women and minorities is a flaw that could severely harm the scientific society in America. Virginia Woolf once said, "There is a spot the size of a shilling at the back of the head which one can never see for oneself." Truly, without the presence of underrepresented groups in the scientific world, it is never possible for the scientific world, which has been dominated by males, to truly see the full perspective, given the fact that female scientists have also contributed greatly to the scientific world. In conclusion, taking another look at Cobb's life, she not only struggled through these filters herself, but also broke these filters for scientists from underrepresented groups who are following her steps. Thank you.